not necessarily represent the opinions of Revolution Radio and FreedomSlips.com, its staff, or affiliates. You're listening to Revolution Radio, FreedomSlips.com, 100% listener-supported radio, and now we return you to your host.
You're back with Dave and Mecky's Shiny Side Out, believe it or not. That's where you are. I'm seeing if I can work the panel. You're listening to the number one listener supporter talk radio in the net, so push the donate button and subscribe to the archives for free. Believe it or not, they're free. Mecky, if you're still with me. I don't know where you are. So, get yourself some great merchandise from the station's website, a t-shirt, a mug, or anything you like. And thanks for listening to Revolution Radio. This is episode 691, and we are talking about human-animal hybrids. This is part 2 in the series, so do catch up if you don't know. Let me see if I can change that. Oh no, I think I can kind of leave that there. Let me see. No, I don't know. I can't see me. Oh, I see what I can do now. Okay, well that's pretty cool. Let me work this out. Mecky's going to come back with his video in a minute, no doubt. I'm going to open up, since I've done the show notes. Let me have a look. It takes a second for me. I'm sure we can sort this out. Give me a sec. Hopefully you can hear me in the chat room. Can you tell me if you can hear me just fine? And I realize there's a 30-second delay. Let me have a look at this. Oh, it opened up. Oh, that's right. It's weird. Teams opens stuff up in my cloud, so I've got to go to a different place and see if I can get it from there. Hang on 1 second. He sent it to me last week. Here we go.
Everything okie dokie?
I'm here. Are you there, Mecky?
I'm here, brother.
Oh, good. I've read us in. I've worked out how to do the music and see the music, by the way. So
I can't not.
No, you can't. Don't worry. It's okay. Let me see if I can work out how to have both of us on the screen at the same time. That'd be great. I think because I'm seeing that one.
Things of magic.
There we go. I've sorted it out.
Oh, look at you sorting things out. Lovely. Lovely. Lovely.
So, it's weird, Mecky, because my OneDrive.live.com opened up the show notes from last week, and I don't know where we're at.
Oh, okay. Oh, my Word conveniently has frozen. There we go. That's better. Maybe not yet. No, we're getting there. We didn't get far into the text, but suffice it to say, we did talk about CRISPR. So I think what we're going to talk about more is some more detailed understanding of what CRISPR actually is. So let me just get to the right place in the text. So we are talking about human-animal hybrids. That's what we're talking about, just for everybody's renewed attention. We've talked about this. We've talked about that. Here we go. We're actually at the Stanford Explainer. This is an article from June 10th, 2024. CRISPR, gene editing, and beyond. Okay, it's a Q&A with bioengineer Stanley Key, or Chi, on the past, present, and future of CRISPR, CRISPR. And if you remember, we told you last week also what CRISPR actually stands for. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Okay, so over the past decade, CRISPR has taken the biomedical world and life sciences by storm for its ability to easily and precisely edit DNA. Okay, here in this article, Stanford University bioengineer Stanley Chi explains how CRISPR works, why it's such an important tool, and how it could be used in the future, including current developments in using CRISPR to edit the epigenome, which involves altering the chemistry of DNA instead of the DNA sequence itself. CRISPR, he says, this is quoting Chi now, CRISPR is not merely a tool for research. It's becoming a discipline, a driving force, and a promise that solves long-standing challenges from basic science, engineering, medicine, and the environment, says Chi, an associate professor in the Department of Bioengineering and institute scholar at Sarafan CAMH. That's what they're calling it. And he goes on, together we can think innovatively about how to match needs with technologies to solve the most challenging problems. Okay, so as we move into the future, everybody, gene editing, the ability to fundamentally change how, who, and even what we are is going to come into the forefront of our existence. Now, you know, one of the fastest growing branches of medical science or in the medical field, Dave?
No, what's that?
Cosmetic surgery. Okay,
Oh,
it is by
okay.
far one of the fastest growing and most lucrative. In fact, one of the country that has probably taken cosmetic surgery to completely new heights and pinnacles of achievement is not America or California. No, not even Europe. It's actually South Korea. South Korea. If you look at the boy and girl band or K-pop phenomenon of South Korea, you will find that they all look the same. Now, I'm not being racist here, right? The truth is they are all the same because they're all part of a template that a lot of young kids in South Korea are following. They're going to their, plastic surgeon and say, look, I want to look like this. And they all want to look like the K-pop stars. And all the K-pop stars all look like each other. This is a thing. Now, this is an extreme form of plastic surgery. Imagine, Dave, if they had access to CRISPR, where they could alter their genetic code in order to look whatever way they want to look, right? Thailand is another place, interestingly, which has a very high uptake of plastic, surgery. And the thing is this: I think that being able to edit yourself into any shape, color, height, whatever that you want will pretty much, relegate plastic surgery to the sidelines. I think it'll only come into play when it deals with injuries, like serious injuries, facial injuries, etc., where it is all about rebuilding, for example, a face, as it were, right, or things like that, rather than giving you a pout to your lips or a bigger bosom or whatever it is that you went for. So I think it's going, like, plastic surgery is going to go back to where it started in many ways once CRISPR takes off. Did you find the text, my friend?
Yeah, we're at what Chi discussed.
Correct. Why don't we take each question and answer, and then you can start with what is CRISPR if you like.
Yeah, what is CRISPR? So the short answer, CRISPR, CRISPR, is an immune system used by microbes to find and eliminate unwanted invaders. CRISPR, stands for clustered interspaced short palindromic repeats. Biologists use the term to describe the genetic appearance of a system that was discovered in microbes, including bacteria and archaea, as early as 1987. For a long time, no one really understood what it did. But around 2005, researchers discovered CRISPR is an immune system. It's used by microbes to help protect themselves from invading viruses. To stop invaders, the microbes use CRISPR to recognize and eliminate specific trespassers.
How does it work? Short answer. When a virus or other invader enters a bacterial cell, the bacterium incorporates some of the trespasser's DNA into its own genome so it can find and eliminate the virus during future infections. Chi's answer, the bioengineer, it's similar to the human immune system. when a virus infects us, we generate an immune memory in the form of antibodies. Lots of them. Then, when the same virus infects us again, these antibodies quickly recognize the invaders and eliminate them. That's the whole point of a vaccine, for example. When a virus infects a bacterial cell, CRISPR helps establish a memory, a genetic one. The bacterium takes a piece of the virus's genome and inserts the DNA into its own genome. Right? So it actually takes a piece of the virus's DNA and puts it into its own DNA. From that newly acquired DNA sequence, CRISPR creates a new guide RNA, a sequence that helps CRISPR find the invader via sequence complementarity. That is, A binds to T and C binds to G. So the next time when the virus infects the bacteria cell, the guide RNA rapidly recognizes the virus DNA sequence, binds to it, and destroys it.
Number 3, what are gene therapy and cell therapy? And how is CRISPR involved? The short answer, gene therapy can mean using CRISPR as a macromolecule drug to either fix a mutated gene or regulate a defective gene to treat a disease. Cell therapy means using CRISPR to make your body's cells attack toxic cells or regenerate beneficial cells. Gene therapy can mean two things. One is to fix a mutated gene, and the other is to regulate a gene's expression into protein products. Our current understanding of gene therapy is still rapidly advancing, and the challenge is managing therapy safely and cheaply. Furthermore, we're only looking at the simplest genetic diseases. For example, sickle cell anemia is a disease we know a lot about, and it's often caused by a single mutation. So, we can configure CRISPR to fix it, but many more diseases are caused by widespread or more widespread mutations, multiple mutations, and even multiple genes. In the future, gene therapy could go beyond a single mutation, and I'm optimistic, says Chi, that in the next decades, gene therapy will become a pillar of medicine. Cell therapy, on the other hand, is a little different. For example, when people try to treat leukemia, a type of white blood cell tumor, sometimes chemotherapy drugs can completely get rid of the tumor cells. In the past 2 decades, scientists have found that if they retrieve some of the patient's T cells which fight infections, these cells can be re-engineered or engineered as better fighters to recognize and eliminate tumor cells. It's not a tumor. When the modified T cells are injected back into the patient, they can attack the tumors. However, cells are quite complicated. Sometimes they go out of control when injected back into the patient, killing healthy cells along with the tumor cells. At other times, they may fail to work because they are suppressed by the tumor cells. CRISPR offers a powerful tool to enhance the efficacy and safety of these immune cells so that they are completely under our control for best clinical benefits.
How does it differ from other gene editing tools? The short answer is, well, CRISPR is much easier to program than any of the other tools. But this is what she thinks. Before CRISPR, most gene editing tools were a single protein. By changing the peptide sequence of these proteins, scientists could alter their target. To change the target, you need to completely redesign the protein sequence and then test if it even works, which is tedious, unpredictable, and time-consuming. These gene editing tools were theoretically interesting, but they were difficult to use for large-scale studies in therapeutics. Compared to that, CRISPR is elegant because the target recognition sequence is mostly encoded within an RNA rather than a protein, and redesigning the sequence is one of the simplest things you can do in molecular biology. It makes genome editing similar to operating a GPS. If you want to go to destination A, just type the address. And to change to destination B, you just enter the new location. So this tool dramatically reduces the burden, cost, timing, while increasing the precision and accuracy of a gene editing system.
This is another really good question. Why is it such a big deal? The short answer, CRISPR can precisely modify a piece of DNA or its chemistry, so-called epigenetics, in the human body, making it a potential tool for clinical uses in the biomedical sciences. Chi says, CRISPR is a molecule and tool desired by everyone who works in the life sciences, biomedical research and clinical settings. Its high precision is unparalleled and enables many uses, including gene therapy. My dream, says Chi, has been to develop new biotechnologies and apply them to diseases without a cure. Finally, Mekki, an actual cure objective rather than treating the symptoms. The Simpsons. Yeah. Genetic diseases make up a big part of this category. Traditional medicines, small molecule drugs, surgery and other methods don't work for these types of diseases, but CRISPR molecules have become highly promising as treatments because they allow us to precisely modify a piece of DNA in the human body. This could lead not only to relief, but also to a cure. Indeed, recent FDA approval of the first CRISPR drug, Casgevy, in treating sickle cell anemia and beta thalassemia, that'll do years, speaks to its safety and potential for other diseases. Sickle cell anemia is a disease in which people have a mutation in their blood, in their red blood cells. Normally, there's no treatment other than frequent blood transfusions or bone marrow transplants from a matched donor, which are expensive and damaging to a patient's overall health. Using CRISPR, it's possible to perform a one-time treatment to permanently correct the mutation. There are more than 8,000 genetic diseases like that which can be potentially considered.
That's a huge potential load that we can fix here. If you think about it, massive. Now
I mentioned my daughter has a mutation on one gene only, and it would be possible to have corrected hers rather than surgery.
Yeah,
but we're not there yet.
Maybe. Well, yeah, but I don't think we're too far away. I hope. Now, how far has CRISPR technology come since it was created? Short answer: in about a decade. Remember, this is the 2024 article, right? Mm-hmm. In about a decade, scientists went from wondering if this technology would even work in human cells to getting the first CRISPR drug-approved uses in the clinic. Mm-hmm. So the first use cases have been approved. So this is what Dr. Chi. That's what he thinks. In 2010, I was working on CRISPR as a bioengineering graduate student at the University of California, Berkeley, under Adam Arkin, a synthetic biologist and bioengineer, and collaborated with Jennifer Doudna, a biochemist and structural biologist. In the early days, CRISPR's practical usefulness was not very publicly recognized. At that time, many counter-arguments said that CRISPR was just a bacterial system, and most of these simply don't work in human cells, which, to be fair, is true. But after Jennifer Doudna and Emmanuel Charpentier published their seminal 2012 paper on Cas9, one type of CRISPR that cuts DNA using a single protein and an engineered single guide RNA, the research and published papers grew exponentially. Firstly, because it's a system that everyone in the life science wants. Secondly, using CRISPR is super easy, flexible, and robust. It's not like other technologies that take multiple years and billions of dollars to set up. CRISPR only takes a couple of weeks and a bit more than a few $100s to set up now. Sorry, I'm going to repeat that, okay? CRISPR takes a couple of weeks and a bit more than a few $100s to set up now. That's phenomenal if you really think about the implications here, right? A lot of researchers significantly contributed to the rapid development. For example, within 3 years following its initial demonstration, structural biologists solved the high-resolution three-dimensional structure of what Cas9 and other CRISPR proteins look like. Bioinformaticians, I didn't know that was a thing, have revealed many new species of Cas9 molecules beyond Cas9, many of which have novel functions. Biochemists engineered CRISPR to understand how fast and tightly it binds to DNA. Bioengineers, including me, that is, Dr. Chi, of course, engineered the proteins to make them work more efficiently and more specifically so they can work better in the human body for gene therapies. Also, clinical researchers started to use the tool to address particular diseases. Furthermore, the applications of CRISPR went beyond gene editing. Epigenetic editing is an exciting development, although we still await clinical benefits. It was used for targeting the human three-dimensional genome, visualizing the DNA dynamics, or even targeting another set of molecules, RNA, for gene regulation. I don't think I'm exaggerating, he goes on to say, that essentially CRISPR has been tested as a potential treatment option for every disease that we have clear knowledge about. Let me say this again. CRISPR has been tested as a potential treatment option for every disease that we have clear knowledge about. CRISPR can't solve all of them, but because this tool is so powerful, easy to use, and so far-reaching, it has allowed everyone to combine their expertise with CRISPR.
It's unbelievable. So I remember the conspiracy world had been talking about CRISPR since the release of the Cas9 paper. And when I was speaking to my daughter's doctors about CRISPR and the opportunities, they stared at me blankly. And I go, well, look at it up online. And then in the next iteration of the meetings, they were like, wow, that's really good, but we're not there yet. And I go, I know we're not there yet, but I wanted to let you know that this is one of those things that could have been rectified by it. And then we had COVID, and Beam and Pfizer got together and started their own for a couple of $100, Becky. They started their own in only a few weeks, and look where we are today. So very important. So thinking about this parallel timeline. This is number 7. In 2019, Victoria Gray was the first person in the US to receive CRISPR treatment for a genetic disease, sickle cell anemia. Now, CRISPR-based therapies are approved in the US and the UK. What is next? The short answer, this is very exciting. Future CRISPR drugs will address more incurable diseases, which provide a test case for CRISPR's efficacy and safety in different organs and patients. Chi says, I'm super excited to see CRISPR becoming a drug to treat a disease as a one-time cure. When CRISPR first came out, there were concerns about whether these bacterial molecules could be used safely in humans and whether it was safe to cut and edit human DNA. Thinking about this as my edit, while people are alive, while they're still functional. It's like changing the wheel of a car while you're driving. It's how good this is. While I've seen actually in the Philippines, they get a what's it called? Not a TikTok. What's it called? A tok-tok. And they tilt it up because it's three-wheeler. They tilt it up and change the wheel while it's on its side, driving still. When CRISPR first came out, there was concerns about whether these bacterial molecules could be used safely in humans and whether it was safe to cut and edit human DNA. While there are still questions regarding long-term effects, beyond the period of clinical trials in tested patients, that is, it is very encouraging that CRISPR is safe and effective. The next step is to expand the scope of CRISPR drugs. Medicine isn't made in one day, he says. Different diseases are caused by different mechanisms. There are already more than dozens of CRISPR clinical trials for different diseases in the liver, immune cells, eyes, and muscles. Furthermore, CRISPR epigenetic editing is expanding the scope of disease to treat more types of muscular dystrophy, retina disorders, and brain diseases. Now, this is all about, this op-ed again, this is all about treating things, and this is the evolution we're seeing. At some point in the future, Meki and I guess that we're going to see modifications, willing modifications.
Correct. I think that's. I think in Western medicine with the CRISPR technology, and I think CRISPR is just a start, to be honest. What we're seeing is a move away from managing a disease, right, especially a severe fatal disease like cancers, etc., to curing, actually treating it, not managing, but treating and curing. And in what Dr. Chi here says is cured in one instance. Like you
That's
take
right. Single injection done.
Drug.
Yeah.
One and done. One and done. What? One and done. Now, before I go back to the text, think about the impact this will have on the pharmaceutical industry. Right now, CRISPR is ludicrously cheap and apparently easy. Right? I'm not a biochemist. I'm not a bioengineer, but from everything I've read and seen, including this article here, it is ludicrously cheap. And it's so no longer can the pharmaceutical companies say, oh, we spent billions in research and blah, blah. That's why everything's so expensive. Complete nonsense.
And we need to recoup all those funds.
Not anymore.
That's right.
Not anymore. So
no.
Are we seeing the end of the pharmaceutical companies? I want to say...
I think they'll shift. I think they'll shift and pivot on their product lines.
I think there's going to be a revolution, Dave. I honestly think that.
I think
the reason
so too.
I'm saying that because the only country in the world where medicine is as expensive as it is America, the United States of America.
That's right.
Everywhere else, medicine and especially critical medicine is reasonably priced.
That's right.
Everywhere else, not in the United States. So I think I see a massive revolution potentially in the United States, and the pharmaceutical companies. I can see a lot of them disappearing unless, as Dave says here, they can pivot. If they can pivot, if they can change, and look, the field that is opening up here, and again, to your point, my friend, it's opening up. Yes, we are now treating a disease, we're curing a disease in one go, but now we've opened the door to gene editing full stop.
That's right.
Right? Full stop. Now, what is next? Are we going to engineer and I'm putting this in inverted commas here, in air commas if you can't see the video. Are we going to create the perfect human? What does that even mean? Right? In 100 years from now, if Dave and I, finally test our time machine for the first time, and the last time, are we going to find a world that is populated by 7-foot-tall, red-haired, blue-eyed giants? Right? I don't know. But my point here is, if we give people the freedom, and we have to really be careful here, right? Because once the cat is out of the bag, that's it. But can we give people the freedom to choose exactly what kind of child they would like to bring into this world, or even modifications for themselves? I mean, if it's disease, I think absolutely. I think that shouldn't even be a question. But when do we draw the line, though? And this is really interesting, and I hate to bring Star Trek into it again, but one of the pivotal points in the Star Trek universe's history were the eugenics wars. Essentially, dealing with exactly that question. Are biologically enhanced human beings, genetically engineered human beings, are they allowed to exist? And these are storylines that you find threaded throughout Star Trek. Dr. Bashir was an engineered human being, for example. There's some other Star Trek characters that were as well. Now, is it something that we should allow? and then who should be allowed to do it? I mean, if we allow it for one person, we have to allow it for everybody. How much will it cost? Now, using CRISPR or similar tools, what we find is once we start something in anger and get scale to it, it becomes cheaper, faster, and better. So CRISPR is already cheap, right? And fast. Just imagine something that is even cheaper and faster. Maybe we can take pills, Dave, one day that completely change who you are. Maybe we can take pills one day that when we take them, our kids will come out exactly how we want them to in many ways, right? Long term, of course. Go on.
So what CRISPR is going to trend us into, and that's a perfectly clean society if done correctly. And I mean the elimination of hereditary diseases. The person who's treated for that sickle cell anemia won't pass that on anymore. It won't be in their DNA. So thinking about how that would then go forward, I would imagine that the external perimeter of our breeding pool is going to become cleaner and cleaner until maybe we make the mistake and we can't replicate anymore. Do you know what I mean? There's UFO law bringing that in as a crazy concept, right? But your point. To your point, there is an enhanced Olympics announced. I heard the other day. So sporting events for people who have been banned for performance-enhancing drugs are now allowed to use the performance-enhancing drugs. And it's kind of like the Formula One of humanity, where they're able to compete against each other with those performance-enhancing drugs. Let's see how fast people can run.
No, look, absolutely. It's a special sporting event, and I listened to an athlete that is now going into it. Funnily enough, even with the some of the athletes going into that, even with the enhancements, like we're talking about drugs these days, they're not good enough to compete even in the normal Olympics. So this is like some of the older athletes trying to come back for one shot at glory. But the point that you raised earlier is something I wanted to make a point of as well. This is really, really important, critically important. If we start editing our genes for a certain outcome and a certain template, and maybe at some point, to Dave's point, we'll have a template which everybody desires, right? Or maybe 4 or 5 templates, or 100 templates, whatever. Mm-hmm. But right now, everybody is their own template.
That's right.
No DNA. No DNA is identical to anyone else's DNA. Nobody has that. However, what will happen to our genetic diversity? Yeah. Well, yeah, identical twin, correct. What will happen to our genetic diversity if we do away with all the differences in our genomes? Right now, cleaning it up, of making it free of the disease, I'm all for it, 100%. What we should not do, we should not tailor or edit the genes to fit a certain template, a certain look, a certain whatever. Mm-hmm. Yeah, get rid of disease, yes. Get rid of all the ailments and conditions, 100%. Agree, 100%. Right, but don't create default templates, because we will lose genetic diversity. And then again, you're right. In conspiracy lore, UFO lore, time travel lore, some of the conspiracy theories, some of the theories around this are that, in fact, our descendants have come back through time to harvest our DNA because their DNA has gotten to a point where they no longer can reproduce. And if you think about the stories of the Greys, they're all identical.
That's right.
Now, why anybody would want that as a template, I don't know, but who knows what happens in the future, right? But but maybe even the Nordics, right? Because there are stories, even when the Greys are involved, the people that are abducted and have encounters of the 4th and 5th kind, like intimate encounters, they say that these humans they have them with actually, human in many ways, red-haired usually, and fair-skinned. But those are the ones they have these encounters. Not the Greys. They don't have any relations with the Greys. They have relations with these other humans somehow, right? I don't know how that would work. Maybe there's some kind of highly advanced biological android or something. But my point is, if we don't watch ourselves, right? Because it's already started. We've already started to alter our biology. We are full of micro and nanoplastics. We're full of forever chemicals. We've now started down the route of allergies, like we discussed in the first hour, and all kinds of adverse reactions to our environment. So we've gone down a route where we are now affecting our physical and mental well-being directly through the way we live. Yeah? And if you add to that the potentialities of CRISPR and other gene editing tools, and look, I didn't until I started looking at this in some anger, when we first did this show, and now again with this updated article here, I didn't realize how easy it was and how cheap it was. I had no idea, but it is easy. It is cheap. Of course you would do it. Just we need to be really all these technologies that are seemingly culminating at this point in our world, of which CRISPR is but one. AI is another, energy, and there's a whole bunch of stuff that seems to be coming to a point, a nexus in our development, and we're just not equipped at all to deal with it. Because the thing is this, let's talk about woke just for a second before we finish the questions. Forget hormonal therapy, forget operations. You can gene edit yourself into a woman if you're a man, and vice versa. Eventually, at some point, you will.
I have another argument.
Go on.
My other argument is I take a single hair from the brush that Jennifer Aniston used, right? Grab her DNA and then secretly feed it to my wife, and she becomes Jennifer Aniston.
You know what? With the advancements in robotics, in bioengineering, I can see an entire underclass of, I guess, cybernetic organisms, so part machine, part biology, that will serve our every whim and need. I can see that absolutely happening, that we'll create this underclass of intelligent, not AI, kind of AI, but this intelligent subclass of, I guess, sort of humans, right? That we will be kept as virtual slaves. I can absolutely see that as well. So there's so many avenues where we can get it wrong that we have to walk a really straight and narrow path to get it right.
Oh, the human species doesn't have a big, what's it called? Encyclopedia Britannica of all of our mishaps, has it? No, we haven't. We're fantastic. We'll never do anything like that.
Yeah. Let's just blow it up. Let's see what happens. No.
Given our history, we shouldn't be allowed to use it, to be honest with you.
No, you're right. But of course, now the cat is out of the bag, and that's just how it is. We will use it. Now, okay, let's finish, because I think we can. Let's just finish the article. Question number 8. Were you surprised when the 2020 Nobel Prize in Chemistry went to CRISPR's developers? The short answer, not at all. But I hope the award doesn't lead people to think CRISPR research is finished. It's still growing, and there's much more to explore in basic research, medicine, and beyond. It's the beyond that worries me. Chi thinks, I'm not surprised at all. Even before 2020, researchers had been discussing when the Nobel Committee would recognize CRISPR. So when it happened, I was super excited. Jennifer Doudna, University of California in Berkeley, and Emmanuel Charpentier, Max Planck Unit for the Science of Pathogens, received the Nobel Prize in Chemistry only 7 years after CRISPR was first reported as a molecular system for modifying the human genome. I hope that giving the Nobel Prize to CRISPR won't give people the impression that the genome editing field is done. This is a field that's still growing in every corner of life sciences. Besides being explored as medicine in humans, it is expanding its influence in plants, microbes, and difficult to engineer organisms such as fungi. There are so many questions about how we can use CRISPR for safely controlling the genome, how to use it for novel and innovative research, and how to make it a clinical product that still need to be explored. These are exciting frontiers of further increasing the safety of CRISPR-based therapies and expanding the scope of diseases treatable by this technology. I hadn't even thought, to be honest with you, of all the other organisms we can now
plants and fungi.
Sort of goes into the theme. Yeah, I mean fungi. That's we shouldn't touch them because that's not something.
Good luck
we
with
want.
that. That's not going to end well.
Yeah.
Plants,
Not gonna
I
end well,
swear
my
we'd
friend.
make a triffid in the first 5 minutes. That's what would happen.
Possibly.
It would just uproot itself, walk around. I don't care where you planted me. I'm going over here. I'm not on the clock, am I? I'm going to go take a break. The plants would argue with us, Meki, to be honest. I thought, as soon as you said fungi, I was thinking of how we've got human brain cells on a silicon chip in a robot at the moment. Yeah. Have you seen that? So we could do something to that to make it maybe 64-bit or whatever you wanted to. You could imagine, because I'm thinking in technical terms, but you could enhance that, I suppose. I think the threat is whatever you make, you got to think about if it was like D&D, you're rolling a character, and what traits had you forgotten about? What traits did you not consider in the rolling of your character? If you left one out, you could be danger for you later on. I think the threat of anything we do, and I have to look at it always this, because my brain is wired for worst case scenario observance, at least. And for me, that whatever we do is going to turn out bad anyway. Hopefully the goods last longer than the bad comes around, so we're alive long enough to see the benefits of the whole thing. I think eliminating disease is really important, but all the enhancements is going to I think it's going to change who we are as humanity. We would be the subclass to whatever superhumans were ever made. That if you decided you wanted to make the star skull child, right? If you wanted to make one of those, you wanted to make a human that was 50 times more intelligent than anything that's ever been ever, and you make the neck too spindly, it can only live in space, blah blah blah, and it was smarter than every human ever been, ever, even smarter than AI. If you could do that, and it could live, I'm going to do Dr. Marvin Monroe and say if it would have to live in a booth for its whole life, and I estimate it's going to have a deep resentment against me for creating it. That's what I think we're at. We're at a point where we could create anything, and you have to ask yourself why you would want to embark upon creating anything. Same argument I give anyone who wants a tattoo. Not saying whoever has tattoos is bad or wrong, no. But if you've got a tattoo on your body that you are no longer proud of, that consideration should also be here if you're creating something in CRISPR. And think about what you want, wait a year, do you still think the same way? And the answer is usually no, I changed my mind. But you'll live with the tattoo that you've chosen just for the pure fact that you chose it, not that you still think that way. It's really crazy how this works. Number 9, Meki, before you run out of time, and it rhymes. Besides treatment for diseases, what are other real-world applications for CRISPR technology? The short answer, some other uses are diagnostics, manufacturing, sustainability, and ecological engineering. She says CRISPR can be used for diagnostics. It has been developed as a way to sensitively detect pathogens in the environment that are affecting our bodies. There are also opportunities in manufacturing, such as making products that we care about using organisms like yeast and bacteria. Imagine that we could use CRISPR to engineer new microbes that could boost production, like 10x, more beer, for instance, and also beer that tastes much better and can be catered to different people's wants and needs. Sustainability, I'm going to say that she is a beer drinker. Sustainability is also a big application for CRISPR via bioengineering, creating sustainable, carbon neutral methods of energy or food production is a challenge. genome engineering may offer better manufacturing protocols through microbes that reduce greenhouse gases, plastic, and food waste. Finally, we get to ecological engineering. For example, people are trying to eliminate certain invading or pathogenic mosquito species using CRISPR. That turned out really well. Not West Nile virus, the other one. The what river? The Zika River. What's it called again? Which one?
Ross River.
No, not Ross River. The other one. Anyway.
Which one? The other one.
I forget which one. So, but in my opinion, he says, its long-term safety and impact still need careful evaluation. Other people are trying to revive extinct species. Recently, scientists announced they were trying to revive a woolly mammoth that can live in the Arctic cold.
Yeah, it's always good to bring back extinct species, which goes back to
the Zika virus, Mekki. The Zika virus from the Zika River. Okay. If you remember that, that's where the genetic mosquitoes were released a season earlier.
Yeah, which is Japanese, it causes hydroencephalitis or microencephalitis, I think, in tiny heads, Zika virus. Yeah, look, this is what worries me a lot. Essentially, when you bring back extinct species, right? And how that would then play in anything really, because if you bring back a mammoth, okay, it's a mammoth, who cares? Just not an elephant, right? Same as saber-toothed tiger. But when you start bringing back people, yeah, then you go, okay, I'm bringing back people. I'm not sure that's such a great idea. So is that something we're going to look at? And the answer is yes, of course it is, because we will be able to do it. So that I'm really worried about that aspect, which we'll cover in more depth as the show goes along. And this actually, the next question is perfectly addressing what we just discussed here. What are your views on some of the ethical concerns surrounding CRISPR? And the short answer is, my research group often thinks about the ethics of CRISPR. Some ethically questionable areas are disease prevention and eliminating pesky species, and some definite unethical areas are the enhancement in creating designer babies. And Dr. Chi goes on in detail. The ethical side of CRISPR is something my research group thinks about every day. One of the fundamental principles of ethics is to do no harm. We spoke about this when we did medical experiments. Sure, we want to do something great and helpful to people, but at the same time, we have to consider if we're harming other people. Using that principle, we can consider a few cases. One example is a designer baby, which is a scary topic. That is regarded as unethical because this may create a new human species. When the germ cell, sperm and egg cells are edited, this not only affects that single person, but also the children that person could have in the future. Another concern is in the division of treatment, which has three categories: cure, prevention, and enhancement. Curing someone's disease, of course, is great. Prevention, which means someone is at risk of developing a problem, is a gray area. If someone has a high chance of getting an infectious disease, should we use gene therapy to permanently modify their DNA to reduce their risk? That question really depends on if we have other options. The last category, enhancement, is likely unethical. People talk about the possibility of targeting a gene to grow more muscle or make people smarter or better looking. But if research goes into this category, only some people may be able to afford it. This could amplify the imbalance of socioeconomic status. Another facet to consider is medical necessity. Is the therapy really necessary? Or are there other ways to solve the problem through currently available drugs, diet, exercise, and so on? Beyond medicine, some scientists may want to use CRISPR for ecological reasons. For example, eliminating mosquitoes. From my viewpoint, that's controversial because I think every species exists for a reason. If we try to eliminate mosquitoes, we might have a chain reaction that affects other life forms in the environment and can be irreversible. I hope in the future we can make this technology reversible, like installing a switch, so that if we make something that turns out to be less than ideal, we still have some way to reset it.
Wow, 5 minutes. Number 11, your group demonstrated that it's possible to shrink CRISPR. Why is this significant? The short answer, it's tricky to deliver CRISPR molecules into cells. Shrinking the size of the molecule helps it easily traverse inside of the cells and get to its DNA target. Chi says, CRISPR is such a magic molecule, but that magic only works if CRISPR gets inside the cells and touches the DNA. The question is obvious. How can we even make CRISPR get inside the cell? Human cells are designed to resist any invading DNA. The body has many strategies to prevent foreign DNA from getting in. Many delivery methods scientists use have limited power. We can use retooled viruses to deliver clinical products into cells, but they have a small capacity. The Cas9 version of CRISPR usually doesn't fit inside the virus. Therefore, the currently approved CRISPR drug requires isolating patient cells, modifying them and putting them back in. This process is costly and slow. If we want CRISPR to become a broadly useful medicine, then we need to make the molecule as small as possible. That's why we made this miniature CRISPR, which we call Cas-Mini, which is only half the size of Cas9. We also saw that it is easier to enter cells and works better than other CRISPR molecules because it can get inside more efficiently. This miniature CRISPR can revolutionize the way that we perform editing in the body. Our hope is to address these technical barriers, then test how miniature CRISPR can be delivered to different parts of the human body to treat various genetic diseases.
Indeed. Just one correction earlier. Zika is a forest, not a river. Just very quickly, so we don't want to tell our listeners
Yes,
something.
that's right.
In any event, have I got enough time? How many minutes?
3.
Okay. What is your lab working on in terms of epigenome editing? Short answer, we're trying to use CRISPR to control gene function rather than editing genes to treat diseases. And the long answer, I'm excited about exploring how to treat diseases without modifying human DNA through epigenome editing. It's a different way of thinking about gene therapy. Unlike gene editing, epigenome editing is reversible, safer, and promising for complex diseases that cannot be easily targeted by gene editing. To enable epigenome editing, we developed the first nuclease deactivated DCase9 in living cells to programmably target and control gene expression without altering the DNA sequence. For example, if a person doesn't have enough properly working proteins, we can use epigenomic editing to increase the gene expression over a long term to make more proteins to compensate for this deficiency problem, thus restoring the function to normal in patients. In other cases, someone may have a gene mutation that produces a toxic product, such as in many muscular dystrophies or neurological diseases. Rather than using CRISPR to modify DNA, we can use our epigenome editing technology to permanently silence the gene without modifying the DNA. So they're just turning it off, I guess, right? I'm excited to test this solution in the clinic, as I believe, this offers a safer strategy for treatment without altering DNA.
Absolutely. So I think we'll leave it there in the text, Mickey. And
Yeah, but
we'll see if I've done
a couple
well to
more
get
left.
the break coming in.
See?
It's not here yet. It's still a minute away, but we should be able to hear
Okay,
it
that's cool.
and see it. Can you see the
Cool.
screen?
I can see the screen, but I can't see if there's a break or...
No. Can you see the Winamp screen?
Yes, I can.
Yeah, cool. Well, we should be able to see the commencement of the break. It's actually quite good technology. Now, we hope
really?
that... Yeah, we fingers crossed. We hope that we will be able to make some more reels. And the success of the reels is great. Thank you very much for assisting us. The other thing is that we've enhanced our video quality as well. That's a side effect of the new platform. And some people were saying, wow, that video is so hard, we can't even tell if you're a human or not. So we'll
Ah!
we'll see what happens in this next week. But do catch us for Tuesday and Thursday morning for Plato's Cave at 7 a.m. Eastern US time, and we'll see you again the same time, same channel, Studio A, Revolution Radio, next Sunday.
That's the
And
music.
I can see the music. You can see it playing. See, Becky, look. How good is
I
this?
know it's.
It's great technology. We love it.
It's computers, Dave. It's computers. It's all computer. It is,
Anyway,
it
we'll
is.
see you next week. Take care, everyone.